Right, so hello everyone, I'm Paul, and today we're going to be looking at some exquisitely fossilized polychaetes, as you can see up here. These polychaetes are rather unique in the fact that they preserve almost the entirety of their soft-bodied muscle tissue in three-dimensional white calcium phosphate. And as you can see here, these fibers reach quite high resolution. So, as you might imagine, these are rather unique when it comes to looking at fossilized organisms, and they've really provided an opportunity for us to lift the annelid on fossilization biases, and which has opened a whole proverbial can of worms on this taxonomic pathway. So, but first, what exactly is fossilization? So, fossilization can be defined as the replication and replacement of organic structures and tissues by phosphate group minerals. This often results in high fidelity fossil materials that can and frequently does preserve cellular details. Now, many of you will be familiar with phosphate preservation or fossilization in some form, whether it be the embryos of the Doshanto Formation and the controversies that surround them, to the phosphatized arthropods of Austin. But we're not interested in these when it comes to soft tissue mineralization. What we are interested in is a third type that I like to call Chirin type fossilization. As you can see here, Chirin type fossilization often results in the preservation of myomeres of muscle tissue, normally quite patchy, as you can see in these whitish myomeres here. This isn't actually its tail, this is just someone trying to make a quick buck out of making a nice looking fossil. So, fossilization, this Chirin type fossilization is, whilst we used to not know too much about it, studies during the 90s have revealed to us the manner in which this type of fossilization occurs, and it mainly requires anoxic carbonate lagoons, typical of first the Solnhofen limestone and other localities worldwide, and also the requirement of having microbial mats. These microbial mats overgrow a decaying carcass and create this sealed microenvironment. During this in this microenvironment, decay continues as normal, and because this microenvironment is sealed, it allows concentration of essential decay products. Then, upon reaching a critical pH threshold, phosphate mineralization usurps the normal precipitation of calcium carbonate within these lagoons and allows the phosphate in this environment to rapidly mineralize the creature in question. While we know this much, however, there are quite a lot of things we still don't know, particularly when it comes to addressing biases, such as why do certain organisms phosphatize more than others? Why are there taxonomic biases? How does tissue histology actually influence the process of phosphatization? These things we're not so sure on. So, this is where our phosphatized annelids from the Lebanese Lagerstätte come in. These polychaetes are from the Lagerstätten of Hula, one of the four prominent Lagerstättens of northern Lebanon. They're of late Cretaceous age and have much akin, as I've previously mentioned, to the Solnhofen limestones, here pictured at Eichstätt quarry, these yellowish lithographic limestones, or platencalcs for the Germans out there. These sites are particularly well known for their lovely preservation of fish in these iron oxide stained fossils, and also of arthropods. On rare occasions, however, we get these phosphatized soft tissues. As you can see here, this is often, as in this fish, often inconsistent and poor, and most unusually, it occurs in close proximity to those organisms that also do not bear phosphatized tissues, which makes it an interesting paradigm to study. Even rarer are the aforementioned completely phosphatized polychaetes, which we've only know of about 12 worldwide. There may be another few in the NHM and elsewhere, but they're fairly expensive, so they're difficult to get your hands on. So, this is what our project set out to do. Our first goal was to attempt to identify the biology and map out the myoanatomy and compare that to modern polychaetes to see if there's a biological trigger for this extensive fossilization. To do this, first of all we used camera lucida to obviously map out the myoanatomy and see what exactly we had to deal with, and then we hopped over to the NHM, as you can see from this stock photo that you've probably seen about 4 times today already. But following that, we also looked at the microfidelity of the muscle fibers to see if we can identify any biases at a higher resolution than that, and see if we can further define the process of fossilization within these organisms. So, Overlaying our results of our minor anatomical mapping, there are 4 main muscle groups, the fourth of which unfortunately is not on this specimen. We have in blue here the parapodial muscles, those that make up the fleshy outgrowths of the body wall. In green, the intestinal muscles, obviously that of the gut, and these longitudinal muscles here marked faintly in red, which run anterior-posteriorly down the body. The fourth is circular muscle tissue, which, if we zoom in, we can see here these 2 thin sheets of circular muscle tissue on what we've identified as the ventral surface. Looking again at other parts, we can see a total of 1, 2, 3, 4 patchy and inconsistent of these longitudinal muscle bands along the dorsal surface of the body, and a further 2 larger, much thicker set of ventral longitudinal muscles. Using these features, along with other anatomical features, such as the presence of these biramous parapodia with these cirri structures, bundled ossiculi, these small pseudoskeletal elements, and an unarmed muscular pharynx, which is not pictured here. With all these features, we can identify these as the Amphinomidae, or as belonging to the Amphinomidae, also known as the fireworms. And indeed, when we compare to a CT scan fireworm, you can see a very similar pattern emerging. These, in turquoise here, these circular muscle, 2 sheets on the ventral surface and 1 on the dorsal surface, 2 large ventral longitudinal muscles, and also 1, 2, 3, 4. It's a bit vague, I know, but I didn't have enough time to clean it up, I'm afraid. 4 of these dorsal longitudinal muscles. So, what does this imply? Well, the fact that all of these polychaetes can be identified to this level across them all suggests that there is a taxonomic bias prevalent in this thing, which, while has been noted before by authors, this hasn't been mentioned before at such a small level or such a low taxonomic rank, being effectively within one species within the Lebanese Lagerstätten. So, moving down onto the microscopic level, you can see here this picture again with these well-defined muscle fibers. And if we zoom in on these, we can see that they are composed of individual phosphate grains with a kind of granular texture. Effectively, these phosphate grains consist of many other much smaller phosphate grains tightly fused together, known as microspheric grains. This texture, my bad, varies significantly. While we've got high resolution here, other places you can see that the fibers are very poorly outlined, and this structure is much harder to see. And in other places, it's even granular and crystalline, representing a phosphatization paradigm we're not really familiar with. Also, the presence of bacterial pits, such as this one, the only one of its type found within the fossils, suggests something called an intermediate microfabric, which is bacterially induced. This texture, however, is unusual because it also appears to vary between muscle groups themselves. Here, ringed in red, we have the longitudinal muscles, which you can see are well defined with this microspheric texture, and we also have the parapodium musculature, which is very similar in its shape. However, when we move into the intestinal musculature of the gut, we can see it's a lot poorer, this very moldic texture. There's little of this microspheric texture going on at all, and it's overall very poor. Unfortunately for circular muscle tissue, highlighted green, we are as of yet unsure, though hopefully within the next few weeks we'll be able to answer exactly what the microtexture of this is like. So this also highlights another fossilization bias, which is a tissue bias. The fact that all these 3 tissues have different fidelity suggests that perhaps the histology of the tissues has a strong influence on how exactly fossilization takes place. So, a third, less secure bias can also be identified, a locational bias. As you might be able to, God damn it. What's going on here? It's not going back. Well, that's good. What's being controlled from Tina?
I managed to get to half 4 without a hiccup, so. Yeah, so.
It's not going back yet, no. Odd. Right, if I yeah, thank you very much. Much obliged. All right, so Yeah, you can see that the fidelity of the fibres appears to degrade posteriorly, very well defined here and much poorly defined here. This highlights a third potential bias, a locational bias, but as I'm sure you can see, based upon this small number of samples, this is perhaps not such a reliable conclusion to make. So, 3 biases. First of all, we have this taxonomic bias. This is, as you can see, this unicid here, also from Hewler. It has these very poorly preserved longitudinal muscles, which is quite unlike what we have in these other amphinomid fossils. And while this bias has been noted at much higher levels, normally between a paradigm between fish, arthropods, and coleoids, it's never really been noted at this lower level. We also have this tissue bias between different tissues, perhaps not too surprising given the different tissue histologies between groups. And we have a poorly supported locational biases. How exactly can we explain these then? This is, however, where knowledge about phosphatization starts to fall apart. As you can see here, these are the results of successful replications of soft tissues within the lab via induced decay in anoxic environments. And whilst these are all well and good, whilst this allowed us to actually look at the triggers of fossilization, as I briefly mentioned earlier, but little else was done beyond this. It was kind of just shelved, as it were, as the whole thing being solved. So, but whilst we can't really get any robust explanations for these phenomena, there are a few theoretical theories that might actually help to explain, such as tissue phosphate concentration. It's been correlated quite well that because the phosphate ions utilized in mineralization seem to have a strong control over the extent of mineralization, would it be so unreasonable to assume that perhaps these polychaetes had their tissues enriched in phosphate during life? Again, there's no real support for this, but this is just speculation. Then we have the rate of decay has also been noted for influencing the overall fidelity, and particularly in paradigms against coleoids versus fish, the thin skin of coleoids degrading fast and exposing the flesh faster to decay, resulting in poorer fabrics than the decay through the thick scales of fish. Another third promising avenue would be the characteristics of the musculature, including muscle density and muscle histology, and even chemical composition. It's been shown via unpublished results that cephalopods of the decabrachia have tissues enriched in ammonia, which the alkalinity of which prevents acidity from reaching that critical point that allows phosphatization to occur. What this does highlight on the whole, though, is this huge gulf of knowledge, and we really need further experimentation if we are to truly explore this process and understand what exactly is causing these biases. So, to conclude, we can make 3 conclusions in this study. The white worms of Hewler preserve almost all of their internal myoanatomy in 3 dimensions, allowing robust identification as an amphinomid. 2, that the extent of fossilization has revealed 3 semi-unprecedented biases that lack a really well-defined explanation. And finally, that we require further research to fully understand the triggers of these, and we will effectively require new insights into the process of fossilization in order to answer these questions. So, before I go, I'd like to thank Luke Parry, Jakob Winther of University of Bristol, and also Greg Edgecombe over in the NHM for his help and support, as well as a myriad of other people. Thank you all for listening, and if you have any questions, I'd be willing to take them. Thank you.